Hi, in this video we will be solving question number 46 from GATE 2019 chemistry paper. So the question is the following. The specific rotation of optically pure R enantiomer of 2-bromobutane is minus 112.0. A given sample of 2-bromobutane exhibited a specific rotation of minus 82.88. So the percentage of S enantiomer present in this sample is dash. So in the given question, so the specific rotation of R enantiomer is given, which is minus 112.0, and also the specific rotation of 2-bromobutane in a given sample is also given, which is minus 82.88. Now we have to find out the percentage of S enantiomer present in this sample. Okay, so let's find out. First, let's understand the basics of specific rotation. So specific rotation is the change in orientation of the plane of polarization of incident monochromatic plane polarized light. That means if we pass a monochromatic plane polarized light through a given sample, then if our sample contains a chiral compound, then the given sample actually rotates the plane of polarization. So the degree of rotation of plane of polarization is called or is measured by specific rotation. So here the monochromatic means a single wavelength, not a mixture of wavelengths. So incident light has only a single wavelength. Now plane polarized light means the incident light actually polarized along a particular plane. That means the electric field of the incident light is oscillating along a particular plane. So let's say this is three-dimensional space, Cartesian space. It is x-axis, it is y-axis, and it is z-axis. So let's say our incident light is polarized along yz plane. That means the electric field vector of the incident light is oscillating along that yz plane only. That means like this. This is the electric field vector is oscillating along yz plane. Now if we pass through the plane polarized light of a given sample, then the sample, if the sample contains a chiral compound, then the sample actually rotates the plane of polarization, and the degree of rotation of plane of polarization is called the specific rotation. Now this rotation of plane of polarization is a property of chiral compound, so that's why we call the chiral compound is optically active. Now this rotation can be of two types. One is the dextrorotary and another one is levorotary. So dextrorotary means the compound or given sample rotates the plane of polarization in the clockwise direction. A levorotary means that rotates the plane of polarization in the counterclockwise direction. Okay, now the optical rotation is measured with the instrument called polarimeter. Now for any given solution, the optical rotation is expressed by this equation or expression. Here alpha is the optical rotation. It is written within the third bracket with superscript temperature T. T is the temperature of our solution, and the subscript lambda. Lambda is the wavelength of incident light. Here alpha is the observed rotation, L is the path length of the tube, that means the length of the tube in decimeter unit, and C is the concentration of our solution in gram per ml unit. Now the enantiomeric exchange or optical purity. Now we want to know how much optically pure our given sample is. That means what is the percentage what is the percentage of a particular enantiomer present over the another enantiomer. So so we call it the enantiomeric exchange or optical purity. So the enantiomeric exchange is a ratio of observed rotation and a specific rotation of pure chiral compound. So here alpha is the observed rotation and here alpha is the specific rotation of pure chiral compound. So this is the expression for to calculate enantiomeric exchange or optical purity of our sample. So the observed rotation of a given sample is given in this question, which is minus 82.88, and the specific rotation of a pure chiral compound is also given, which is the R enantiomer of 2-bromobutane, which is minus 112.0. So let's put these values. So enantiomeric exchange is for the given sample is 74%. That means the 74% of R enantiomer is present in our given sample, and the rest, which is the 100 minus 74, which is 26%, is in the form of racemic mixture. What is racemic mixture? So racemic mixture is a mixture containing equal amount of both R and S enantiomer. So 26% racemic mixture means it contains 13% S enantiomer and 13% R enantiomer. Just to take a quick detour, so racemic mixture is actually optically inactive. It actually doesn't orient the plane of polarization of our incident plane polarized light. Why? Because it contains equal amount of R and S enantiomer. So let's say R enantiomer rotates the plane of polarization in the counterclockwise direction, and let's say S enantiomer rotates the plane of polarization in the clockwise direction. So now both the rotation will cancel out each other, and so we don't see innate orientation of plane of polarization. That's why the racemic mixture is optically inactive. So in this given question, the sample contains a racemic mixture which of 26%, which contains 13% S and 13% of R, and 74% R enantiomer is present in our given sample. So the question in the question we have to find out the percentage of S enantiomer present in this sample. So the percentage of S enantiomer present in this sample is 13%. Now for further details, you can visit the following NPTEL reference, which is the NPTEL lecture series on the topic of symmetry, stereochemistry and applications, and the instructor is Professor Anshuman Raichaudhuri from Aizal Mohali. Thank you.